Hello Professor, I am Mubashir Raza. The year of my registration is Spring 2022. I am a master student of molecular biology and human genetics, and my advisor is Professor Ingrid Value. Here is the checklist of my progress report. In current semester, I have registered 6 courses that contain the total credit hour of 6. My timeline. I reached in Sachi University on 25th April 2022. In April and May, I learned about the different techniques like cell culture, pipetting, sample preparation, and genotyping of mice, restraint stress and animal behavior, open field test, and mice handling. In June, I am learning about Western blot. In this experiment, I learned about the cell culturing of 8022 cells, and here is the confluency of 8022 cells after 24 hours, after 48 hours, and at 4 day. I also learned about the genotyping of ATF3 mice. In my experiment, I found 6 wild type mice and 7 heterozygous mice. In this experiment, I learned how to do restraint stress in mice, especially in ATF3 mice. In this experiment, I learned how to handle the mice during behavior test as well as during drugs injection. In this experiment, I learned about the Western blot and protein analysis for sleep deprivation mice. Future direction. I have the project with the title of The Effect of Litsi-Kibiba on Pathology of Alzheimer's Disease in Vitro and Vivo Studies. So what is Alzheimer's disease? Alzheimer's disease is a neurodegenerative disease that has two pathological hallmarks. One is extracellular deposition of amyloid beta plugs and intracellular deposition of tau neurofibrillary tangles. These cause the shrinkage of the cerebral cortex, shrinkage of the hippocampus, and enlarged ventricles in the brain of the Alzheimer's disease patient. Litsi-Kibiba is a medicinal plant and it has antioxidant and anti-inflammatory effects. So for this purpose, I am going to use LC as a medicinal drug for Alzheimer's disease treatment. Hypothesis. LC can improve the pathology of AD in vitro and in vivo studies. For in vitro studies, I am going to use 8022 cells, and for in vivo studies, I am going to use 3xTg AD mice. Aim of my studies. So specific aim 1, here is the experiment 1. In experiment 1, I will investigate the effect of LC in 8022 cells. For this study, I have 3 groups of the cells. One is control, sham, and LC. I will treat the cells in different concentration with LC, and then I will perform MTT assay or ATP assay to check the cell viability. The expected result of this experiment is LC will not show any toxic effect on 8022 cells. In second experiment, I will identify the effect of LC and molecular pathways involved in A beta induced 8022 cells. For this study, I have 7 group of cells up to from 4th to 7th group of cells. I will treat with the LC, and I will perform the MTT assay and ATP assay to check the cell viability. After that, I will perform ELISA test, Western blot, immunocytochemistry for protein analysis. The expected result of this experiment is LC will reduce the amyloid beta toxicity by inflammatory and oxidative stress pathways. Specific aim 2 is about the study to study the effect of LC on learning and memory of 3xTg AD mice. For this study, I have 5 group of the mice. Group 4th and 5th will be treated with LC. Each group consists of 8 mice and having the age of 6 months, and I will perform the behavior test for each group. First is open field test for locomotory functions, T maze for working memory, Morris water maze for spatial memory, and trace film conditioning for learning and fear memory. The expected result of this study is LC will improve the learning and memory deficit in 3xTG AD mice. Specific aim 3 is about to determine the mechanism of LC that regulates the learning and memory in 3xTG AD mice. For this study, I have 5 group of mice. Each consists of 36 mice having the age of 6 months, and then I will perform the behavior test. After that, I will sacrifice the mice and collect the brain tissues and protein from the hippocampus, prefrontal cortex, and amygdala. Then I will perform the ELISA, long-term potentiation, Western blot, and immunohistochemistry for inflammatory markers, oxidative stress markers, and microglial activity markers. The expected result of this experiment is LC will improve the learning and memory deficit by decreasing the inflammatory oxidative stress and microbial activity markers in 3xTG AD mice. Along with this project, I will learn the behavior test like Morris water maze, T maze, trace field conditioning, tail flick test, and pinprick test, elevated plus maze, and passive avoidance test. I will learn also about the techniques like immunohistochemistry, Western blot, and qPCR. In upcoming semester, I will select the course of 8 credit hours to complete my credit hour requirements for my graduation. That's all. Thank you so much.